All right. So before I introduce Aaron, so before I introduce Aaron, next week, we'll on the 13th, we have another talk. It will be Shin, who is going to talk about combating climate change through carbon negative concrete, which I'm really interested to see because I talk to my students about concrete as being one of the major producers of greenhouse gases. And so it'll be really interesting to see that next week. But this week, we are marine fossils this week. So this is Aaron Kilmury. He's been instructing paleontology at Brandon University this term, and he's a former Royal Tyrrell Museum of Paleontology preparation and field technician, a recent Master of Science graduate who studied marine vertebrate paleoecology and biogeography at University of Manitoba, and has recently published the first main chapter of his thesis examining biogeography of marine vertebrates, so fish, sharks, birds, reptiles, all marine vertebrates, of the Late Cretaceous, so 66 to 100 million years ago, in the western interior seaway of North America. That's what you're going to talk about today, mostly?
That is, yeah.
Yeah. In order to investigate the impact of long-term climate changes on marine vertebrate communities in the ancient past. And he's just finished his second main chapter, investigating late Cretaceous fossils of small-bodied vertebrates of Manitoba, with an accompanying open access article that's being published. Nice. His second main chapter also involved a lot of other students. He had who were junior high school students to increase their interest in paleontology and other earth sciences. So I will turn it over to Aaron, and the floor is yours.
Excellent. Thank you very much for the warm introduction, and thank you all for coming. And I want to thank all the staff and students of this faculty for giving me such a warm welcome to the university here. I've been having a great time instructing so far and looking forward to the rest of the term. So thank you all so much for having me. So yes, today I'm going to be talking about my master's thesis research that I completed last year. And I officially submitted this in February of this year. So yeah, still recovering from that. And my main title was Vertebrate Faunal Assemblages of the Late Cretaceous Western Interior Seaway of Manitoba, Canada. And I'm really proud to present this to you because so many times in a lot of different fields in paleontology, Manitoba has been overlooked. So now we get to present Manitoba front and center and show how valuable the fossils are here. So yes, I think I was going back and forth here. The fossils and research discussed were collected from and conducted on original lands of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and homeland of the Métis Nation. So one of the big opportunities of this project was to promote inclusion. It's well known among the sciences, and particularly in the STEM sciences, that Indigenous peoples are not well represented in the STEM sciences, and we really need to put more efforts into involving more communities, especially remote communities, in science research, rather than just doing what's termed parachute science. So one of the things I did with my master's research, as a graduate student, I reached out to some schools and I looked for junior high and even middle school students that might be interested in sorting really small fossils. So one school that was really helpful with this was the Niji-Makwa School in the northern part of Winnipeg that had high enrollment of Indigenous students. And yeah, we were successful in getting some interest in this program. And at this school, they have the Inner City Science Center, which has really nice microscopes for students to use, has lots of great lab tools, lots of good labs that students can really explore what they love about science and get interested and involved in science. So yeah, I encourage everyone to look into the Inner City Science Center. Think about how you might be able to get involved and participate in this. And they also have a website where you can make donations if you're interested in doing that. So yeah, I encourage everyone to think about inclusion and involvement in your projects. So what I have to discuss with the research project today, I split into three main chapters or three main parts. So we'll introduce the Western Interior Seaway first. I'll introduce this project, and the first part is talking about biases. Before we start making interpretations on the data, it's important to look at the results and see what are the main biases that are affecting our interpretations. So there's quite a few of those to touch on. So we'll start with the biases first. The second main chapter is one of my favorites. It's looking at the biogeography, looking at the community zones of the seaway. And the third part almost brings it full circle and tries to address some of the biases that were identified in the first part. And lastly, I'll conclude. I have a couple slides concluding each part of this big research project. So the Western Interior Seaway, what is it? This was a large body of water that was on the surface of a continent, so it's termed a seaway. And it was tropical to subtropical and contained a variety of marine life. So this stretched from what's called now the Arctic Ocean to the modern day Gulf of Mexico. It was a very, very long seaway that was kind of north-south, that stretched across the North American continent. And yeah, some of the life that this seaway contained was microscopic. We have diatoms, radiolarians, and coccolithophores, to name a few. We have macroinvertebrates, like we have some bivalves, we have belemnites. Here we go. Here's one genus called Baculites that's quite common. And there are also ammonites. Here is a common ammonites genus right here, Placenticeras. And there's the vertebrates as well, which you may have heard of, and there's quite a few that are extinct now. But some of the vertebrates are fish. There's lots of fish in the sea. There's turtles, small and large. There's also sharks, rays, and skates that are not shown here. And we have birds. We have some very interesting birds. Many of them still have teeth in their beaks. So we have some very interesting bird families. So that's the closest thing we'll get to dinosaurs in this presentation. There's no traditional dinosaurs that we have in the seaway, although there have been places in North America where dinosaurs have been washed out to sea, and you can find their remains on seafloor environments. Some of the largest animals that we have in the seaway include pliosaurs and other plesiosaurs, short neck and long neck varieties, and the mosasaurs, one of my favorite animals here. So shown on the left is Tylosaurus. That's one of the largest genera that we have in the seaway. And there are some slightly smaller ones, but still very large. There's Plioplatycarpus here, and there's many others. So there's a whole family, a whole order of these animals that are quite unique. They're extinct. They went extinct along with the dinosaurs at the end of the Cretaceous. And their closest living relatives are snakes and monitor lizards like Komodo dragons and marine iguanas. So where this project began is with the research of Dr. Elizabeth Nichols and her PhD advisor, Anthony Russell. So they had, Dr. Nichols had published her PhD thesis in 1990, and she was the first one to analyze community zonation of vertebrates of this Western Interior Seaway. So two zones are proposed, a northern zone and a southern zone. So what was unique about the northern zone was that it had low diversity in all the represented groups, and it had a higher proportion of birds particularly the hesperornithiforms, and these reptiles, the plesiosaurs. So these are the short-necked plesiosaurs that were more common in the north. There we go. And there was also more of this one genus of mosasaur called Platycarpus. The southern interior subprovince, or the southern zone, was characterized by having higher diversity in all tetrapod groups represented there, as well as much more abundance of turtles and sharks. And the specific type of mosasaur that was more common in the south was the genus Clydastes. So Elizabeth Nichols and Anthony Russell, what they were doing with this zonation was trying to look at one specific time period. They're trying to analyze community zones for just 80 million years. So they were using the way they were classifying the age of the rocks were using small fossils that were really good at putting age assignments to rocks. So they were using biostratigraphy, as well as some minerals that could be dated just on. They were looking for radioactive crystals that they can get an absolute age from. So at this time, there was only 2 sites at around this time period that had absolute age determinations. So there was the Manitoba site and sites in South Dakota and Wyoming. So they could be correlated pretty accurately. Although the sites shown as circles here, they had to rely on those smaller fossils that hinted that the age was close, but it wasn't as reliable as having those absolute ages from the crystals. So they correlated all these sites, but we now know that the sites shown as circles were actually a little bit older. So they were close in age, but slightly older. So I wanted to reassess these community zones for this time period, see what is it like with the more fossil data that we have now and more recent age determinations. And I also wanted to assess this for other time periods. I wanted to see, how do these community zones change over time, and how do they respond with changing climate? Okay. So yes, we over the last 20, especially over the last 10 years, we have many more and much more accurate radiometric age determinations using uranium and lead systems, as well as potassium and argon. We can get more accurate ages from some of these beds. So this allows for more reliable correlations of fossils, fossil assemblages, and opens many doors for current research. The Late Cretaceous fossils of Manitoba have potential to answer very large-scale questions because of a few biases that we'll talk about soon. But one of the big reasons that Manitoba is very important is because of its position. It's more north than more of the more sampled sites to the south, like Kansas and South Dakota and Texas. So yeah, we're in a good position to present some very valuable data. And we can assess the biogeography of the Seaway. So the Manitoba escarpment, you may have heard of this term, you may have not. This is what it is. So here is a topographic map showing the Manitoba escarpment through most of Manitoba, as well as a little bit of North Dakota and a little bit into East Central Saskatchewan. So one of the main research questions I have here is, what are the implications that Manitoba fossils have in regards to understanding the biogeography across the entire continent? And how does this show changes in biodiversity over time? And just as importantly, what are the biases that are associated with these fossils, and how can they be appropriately addressed? So here I'll just go over the objectives of each part of this huge project. So the first part is looking at the biases. And these results were published in the Manitoba Geological Survey open access article. So this was mainly a review of museum collections. See, what do we have in museums from Manitoba that are Cretaceous in age? So we did this review to assess the existing biases and then recommend ways to counteract these biases with future work. The second part, these results were published in an online article through the Paleo 3 journal in 2022. So the goals of this chapter were to assess the changes in diversity, as well as what animals were present in certain areas, assess those changes over time, those main changes in the communities, and determine how did the zones change? Were there just 2 zones throughout the whole Late Cretaceous time? Or were there 3 during some times, or even 4? And how did the shape and the size change? And then we can also examine some preservational biases, so we can focus in on one type of bias. The third and final chapter was looking at the small animals. So one of the main biases that were identified was a size bias. Lots of people, when they go to museums, they want to see the big dinosaurs. They want to see the big fossils. So traditionally, that's what people collect. They go looking for the big Tyrannosaur skulls and the big fancy fossils. But that's led to a really pronounced size bias where we actually don't know a whole lot about the smaller fossils. So in this chapter, I look specifically at the smaller fossils, and I look at a rock formation that hasn't been as well sampled. So I try to address some biases in this chapter. Okay. So for the first chapter, here are some results showing some museum survey results here. And what I have shown is museum collections on the x-axis in order from most fossils to least fossils. And then I have them color-coded by rock unit, where we have the oldest rock unit on the bottom and the youngest on top. And yeah, their total numbers are also listed on the top here. And they're also separated by major animal group, where we have fish, birds, sharks, rays, and skates, and traditional reptiles. Okay, so some of the biases that were identified, well, there was quite a few. One of them was preferential sampling of certain geologic units. Because of economic activities in the past, in Manitoba, bentonite used to be mined here. And bentonite, it's a clay that used to be volcanic ash. So across North America, we have many volcanic ash layers that show how active volcanoes were along the Rocky Mountains and the Cordillera to the west. So there was some pronounced intervals of lots of volcanic eruptions and volcanic activity. And there's even some very, very thick volcanic ash layers that make Mount St. Helens look like nothing. So this volcanic ash from about the 1930s to 1990s was mined in Manitoba, and it was kind of used as a filter for a lot of things like winemaking, toothpaste, even kitty litter. Bentonite was widely used. And one of its main uses is in drilling mud for oil drilling, because it's a non-swelling clay, or there's many non-swelling varieties. But anyways, because of the bentonite mining, there was a lot of bentonite mining in the purple unit, the Pembina member. So we actually have a lot of macrovertebrate fossils, mostly birds and reptiles, from the Pembina member because of all this mining. There's also been one limestone layer found in the oldest unit, the Belfourche member, with lots and lots, thousands of shark teeth. So because paleontologists know there's lots of shark teeth in this unit, they keep coming back after a few years and keep sampling this. So we have a large sample of mostly shark teeth from the Belfourche member. So there's been advantageous sampling of certain geologic units and certain animal groups. There's quite a few paleontologists that like to focus on sharks. So we have a good sample of shark teeth. But there's fewer that study fish. So there are lots of fish fossils here. We just need more sampling and more research to be done on them. So yeah, there definitely has been more sampling of sharks, birds, and reptiles in Manitoba. And I already touched on this, there's also larger size classes that have been sampled for the most part. Okay, so for the Nichols and Russell study, I'll just point out a few main differences between them, between that study and my current study. About 5 localities were used in this study, with South Dakota and Wyoming grouped as one. And they were aiming to assess a time interval of about 80 million years, or a narrow time interval that is that old. And they used approximately 5,000 museum specimens to assess these zones. For this study, we assessed a much larger time span. We used several time intervals instead of just one, and many more localities. So there were some localities where only a few sites were the same age and could be used to assess community zones. And yeah, some time periods where up to 9 localities could be used to assess community zones. And there's over all of the time periods that were analyzed, almost 60,000 museum specimens were used to assess these changes in community zones. All right, so I'm just going to check how we're doing on time here. Okay. So there's 2 ways that I calculated community similarity. The first one is the more preferred method. And what this does is it compares the fossil or the taxa lists of each site. It compares the fossil lists with each site simultaneously at the same time. So you're comparing every site with each other by taking subsamples and repeatedly sampling each site to see which sites are most similar and which ones are most dissimilar. So this is done in RStudio using our coding language. The second method is a little bit easier and it's less preferred. What it does is it compares all the sites with one site in particular. So where I had enough sites and enough fossil data, I was able to do the more preferred method on the top, the multi-dimensional scaling. But for most time intervals, there were some sites that had smaller sample sizes, and sometimes there weren't enough sites. So I had to rely on pairwise calculations here. So this was done using Sorensen's coefficient of community, where you compare the proportions of certain species and certain genera to those at other sites. And the percent similarity analysis. So yeah, this uses the proportions of certain taxa, compares them across sites. So most of the images that I'll show you soon are based on this last method on the genus level of fossil data. So this is the one time interval where I had enough data and enough sites to do the more preferred approach, the multidimensional scaling. So what we're seeing here in this ordination space is all the numbers represent different sites across North America. And it compares the ones that are nearest to each other or the most similar to each other. So what we have is we have this kind of triangle, this triangular shape here. And each endpoint is supposed to be the most dissimilar from the other endpoints. So one of the endpoints that makes sense to see is the Northwest Territories. It is the northernmost site, so it should be more dissimilar from the other sites. And the results go pretty closely with Elizabeth Nichols and Anthony Russell's results. where they show the diving birds and the short-necked plesiosaurs are much more abundant to the north. And one of the more central localities in South Dakota, this is characterized by having higher abundance of turtles and one genus of mosasaur. And then another endpoint is one of the southernmost sites down in Texas. And it had much more sharks than other localities. So that fits with the previous study as well. Okay. Some of the interesting pairings here make sense as well. Saskatchewan and Wyoming sites are pretty close to one another. So they were also similar in the types of fossils that they had. As well as Alberta and North Dakota. And then one unusual pairing was Manitoba with Alabama. So they're not at all close to each other. So it was interesting to see them plot closely together on their fossil data. But we think that biases are really influencing this result because what you can see is the size of the circles represents the size of how many fossils are known from that site. So at this time period, one of the youngest time intervals from about 71 to 77 million years ago, Manitoba, we don't have a lot of fossils from this time period. And same with Alabama, there's not many fossils known. So yeah, there's not enough fossils to really show that relationship well. And what they do have is a common fish, which is influencing this. So yeah, there are some biases that you can see influencing the results, but for the most part, these data make sense. Now, that was the one time interval that I could show multidimensional scaling results. For the other time intervals, I had to rely on the pairwise calculations where we compare all the other sites with Manitoba. So here's showing most of the results. So it shows all the fossil assemblages, the ones that were time equivalent or similar age for each time slice. And it shows changes in community zones over time. So that kind of answers our main research question. So what we see here is that there's at least two, a north and a south community zone throughout the late Cretaceous. Although what's interesting is that throughout some of the warmest intervals in Cretaceous time, we see at least 3 zones. So zones are getting smaller, and there's more community zones during the warmest time intervals. So here's a global temperature reconstruction curve from Cretaceous time. And this was published by Scotesi and others in 2021. Shown in the blue box here, this is the time interval that was analyzed from about 98 to 71 million years ago. And the warmest time intervals we have shown in the box labeled B. This is the first warm time interval that kind of lines up with right about the peak of this thermal maximum. And the second warmest time interval that shows 3 community zones labeled in the box labeled F there, is also shown kind of when it's still quite warm. And the reason that we don't have 3 community zones shown in the boxes in between is because we don't actually have any fossils known from the Northwest Territories at this time. So we need to do some more fossil sampling to see if there are 3 community zones that persist throughout this entire warm interval. So one of the preservational biases that we can look at using these data is what I have shown here is all the fossil assemblages or all the sets of fossils ranged by state, province, or territory. And it shows their rock type on the x-axis here. So these are sandstones, mudstones, kind of chalky mudstones, and going all the way up to limestones. So it's arranged from a low amount of calcium in the rock to higher amounts of calcium. And on the y-axis, we're seeing how similar these fossil sites are with Manitoba. And the color represents time intervals. So what we can see from here is that fossil sets that tend to be more similar to Manitoba, go back to this, tend to be calcareous. So most of them have higher proportions of calcium in the rock. And the time periods where we have more calcareous rocks are during the warmer intervals, from late Cenomanian to earliest Campanian time. So we're seeing during the warm intervals, we're seeing better preservation of fossils. We're seeing more complete representations of communities. So our data is a little bit skewed here. We're getting a clearer picture for the warm time intervals, but we're not getting as clear of a picture for the cooler time intervals, especially near the latest late part of the Cretaceous. So it's good to be mindful of that bias. So yeah, talking about biases, hopefully I'm still good on time. So what I did for the third chapter was trying to address some of these biases by looking at the size bias and the geologic units bias. So the Pembina member, the black beds, is where most of the mining happened in Manitoba. So we have a good sample there. And that one limestone bed with thousands of shark teeth, that's down, I think it's just below, or it's kind of near the exbentonite here. So that's down here. It's about 95 million years old. So I wanted to sample some units that were relatively less sampled, and units with really good preservation too, so that we can get a good image of these communities. So I sampled the Fabel Formation. I chose that unit, and I wanted to sample a lower, a middle, and an upper horizon, and compare the small community members. See what the little community members were looking like there. One of my sites was in the Morden, Miami area, and most of my sites were near Dauphin. So yeah, I got to see some really beautiful areas and go along some creeks and sample some very nice outcrops around Dauphin. So let's see if this works. Here's a video showing one of my limestone samples dissolving in an acid bath. Excellent. Okay. So this is from the topmost horizon, the Marco calcarenite. And it turned out to be the horizon with the most fossils. So what we see, all the dark little bits that are black and brown, these are all fossils. So it's just loaded with fossil fragments, mostly fish scales, ribs, and fish vertebrae, but also shark teeth, really interesting bones from the tail fins of fish. There's some really interesting forms. So there's lots of fossils in this. What I did was I digested the limestone and sorted through the remaining material that had little fossils and quartz grains as well. Okay. So there's one of my volunteers. He's an undergraduate student in the geology program at University of Manitoba. And yeah, we spent over 420 hours sorting, identifying, and imaging these little fossils. So we spent a lot of time in these labs working with these tiny fossils. We discovered probably even higher number than this, probably closer to 2,000 fossils that we found, collected, and described. And what was really exciting was that there was about 15 species that we had no idea were in Manitoba before this project. And now we have a much larger record and a better idea of what this community looked like. There's also lots of fish vertebrae, and I found a good use for those. So I measured a lot of vertebrae. That was very hard to do with the little tiny digital calipers underneath a microscope, trying not to break them. So that was a lot of work, but it was very much worth it. We were able to use these images, use these descriptions, and create a student fossil sorting guide. So people, even in middle schools and high schools, if students and teachers are interested, they can use some of this material, sort through it as a class, and discover new fossils for themselves right in the classroom, and see what they are by looking at this guide. So this is what the guide looks like. We just finished that, I guess in the spring. Yeah, I think we put the finishing touches on it this spring. It's online. I have a copy. It's freely available. If anyone's interested, I'm happy to provide it. So I'll just quickly go over some of the interesting little fossils that we found. Here's one of the mystery ones that I was hoping to describe as a new species, but it was too incomplete. We only had fragments of elements, and we didn't have enough complete material to describe this as a new species. So we're still on the lookout for more bits that belong to the same species as this. We're thinking it might have something to do with the fish fins. So it might be some unique bony element in the fish fins, one of the fins. But when I first saw it, I thought it looked like these ancient swords, these Indo-Persian blades called shamshirs. So yeah, I use this as an excuse to go and look at ancient swords and museum collections. Had a great time doing that. And yeah, these are shamshirs from they're held at the Royal Ontario Museum, with gold inlay. Some of them are serrated, have serrated edges. Really cool blades. So yeah, if I end up finding enough complete material, I hope to name it after this really cool blade. Yeah. Some other material that we're able to identify a bit more easily were, yeah, some of these really tiny teeth from these skates called rhinobatos, or they're also called guitarfish. So these are very well preserved because they're covered in enamel. They have a very good, easily preservable tooth. And what really got me excited, what I found was interesting, is we found rostral teeth from sawfishes. We actually used to have sawfishes swimming around here in Manitoba. We also found some really interesting looking fish fossils. This may actually be from several different species in the same family as this fish. So yeah, we might even have more species that we didn't know about here. But we have several jaw elements. Yeah, we have, let's go back a second. These are the lower jaws, upper jaws on the left, and some palatine tooth plates here on the right. These are not microvertebrate fossils. These are some macro ones. So these were surface collected and not put in the acid baths. But these are some larger fossils that were found by a private collector that lives just near Brandon here. And yeah, some of these were donated to the Manitoba Museum so that we can include this in the research. And I'm really grateful for that because yeah, that means more species were recognized from this time period. This one is interesting on the right. It is known from a very blunt-nosed fish, and it was only recently described from Texas. That's where the only occurrences were known from. And now we have at least 3 occurrences here in Manitoba. So there's this really interesting fish that we've really extended the geographic range for. So that's very significant there. This fossil in the middle, this is, we think it's the oldest known mosasaur fossil in Manitoba, and it's about 92 million years old. So that was found in the Marco-Calcarenite as well. And here's an image from the most recently published article that we put out this year in Peer J, describing the small fossils. So we were able to reassess community zones for the Fabel Formation equivalent fossils and see what sites are most similar to Manitoba. And yeah, this really increased the similarity of most sites with Manitoba. Now that we have a better idea of the community here. So yeah, the areas in dark blue are most similar. Okay. Let's see. Just going to go a little bit past this. This is using the vertebrae measurements to see how close the site was to the shoreline during that time. So yeah, pretty much I was able to see that the topmost horizon was the most shallow and most closest to shore because of how poorly sorted the vertebrae were, the amount of quartz that was eroded from the Precambrian shield to the east. And the middle horizon had almost no quartz grains, very few or much less vertebrae and smaller sizes. So that was a bit more offshore. And then the bottommost site, right at the base of the Favel Formation, it had almost no vertebrae, maybe about two very poorly preserved vertebrae were found, no quartz grains, it was all clay. So we thought this was the most offshore. So we can see this change in sea level very clearly from the bottom, middle, and top of the rock formation. And that's representative of the Greenhorn marine cycle. So to conclude, I hope I'm not getting over time, but to conclude, there is a big size bias that we need to address to get a better image of these communities and to get a better interpretation of these community zones. We can see from the current data that we have a much clearer picture of what these community zones look like since 1990. And we can see how they changed over time, but there's still a lot of work to do. And looking at the small community members, it has so much potential for discovery there. It's very valuable information that can give us distances to shoreline, how similar these communities are with other areas, and how these communities respond to major climate changes. So yeah, what I would like to do in the future, hopefully one day I can begin a PhD and go up to the Northwest Territories and try to tackle more of these biases, sample more northern sites, and see see if this hypothesis holds up, where the warmest time intervals have 3 or more community zones. And here's a cast of a fossil from the Northwest Territories. They have beautiful fossils. So I think it's worth going up there to see, go find more. And that is it. Thank you all so much for listening. I hope you found this interesting and I hope you learned something new. Thank you very much.
Thank you very much, Aaron. You're sure that was just a master's thesis?
Oh yeah, I know, right? Yeah.
I've seen less work in PhD dissertations, I think, and that's amazing amount of work. Amazing amount of work.
Thank you so much.
And very interesting. Do we have any questions? If you have a question, put up your hand. I'll bring you the little microphone and you can speak into the microphone. Any questions for Aaron?
So I'm curious about these. It's plesiosaurs up in the Northwest Territories, and they're like super big up there, or bigger than anywhere else. What's that about? Why do they get so big up there? Yeah, I'll leave it there.
Oh, sure. Yeah, great question. Yeah, as far as I know, I think most of the plesiosaurs from the Northwest Territories, there's 2 main groups of plesiosaurs that we know of, the short neck ones and the long neck ones. And there's very few specimens of the long neck ones. So we still have lots to learn about them, about their distribution and their biology and everything. But the short neck plesiosaurs, we have a larger sample of them. And yeah, there's lots more in the Northwest Territories. There's even lots in Saskatchewan as well. And there's some hypotheses that have to do with birthing grounds where they're like some birds as well. They have birthing grounds to the north. So yeah, it might have been better conditions for raising young. I don't know about the maximum size. Wish I had an answer for that, but yeah, it could have been prey items, just more prey items in that area. But yeah, lots of great questions there. Yeah.
Was it cold enough that it could have been thermal regulation?
Could have been, indeed. Yeah. Yeah, these were like really hot time periods. So it does make sense that especially the traditional reptiles were kind of being pushed to the north just to survive. and in some of the seasons too, it would have been extremely hot. So yeah, it's lots of good things that we don't know a whole lot about right now. Lots of things to explore there for sure.
Just curious, when you say you're imaging stuff, how do you go about that? Like, do you take photos, or do you have, like, for example, let's say I found a tooth. I wouldn't know whether it's, species A or species B. So how do you match that? Do you have like a repository or some kind of matching algorithm? I don't know.
Oh yeah, excellent question. Yeah, for some of the fossils, one of the main resources I used for identifications was just articles that were published mainly from Kansas, but also South Dakota and Texas. There's some really good articles describing shark teeth. That's probably one of the most described groups of animals from the sea is all the sharks. And yeah, there's some on the fish, but there definitely needs to be more on the fish. I was trying to identify some fish skull elements, and I just found it nearly impossible for a lot of the families. So yeah, we need more descriptions on fish for sure, more complete fossils. And the way I imaged them was using, I used this special microscope that my advisor ordered right at the start of my program. She ordered this very nice microscope, very tall, that was built for taking 3D stacked images. So we did image stacking for a lot of these tiny fossils. So I'll go back a couple slides. So this was taken using image stacking, but it was one of the largest small fossils. And for the larger fossils, it takes a couple hours to build one focused image out of this big thing. But for most of the fossils, yeah, it was pretty easy, just maybe 15, 30 minutes. You can get a really nice image using image stacking here. But yeah, just took images under a microscope.
Is there anything known about why the biodiversity was so high in the seaway? Was it shallower and warmer? Was the salinity different from the north to the south?
Oh yeah, great question. Yeah, I think there would have been more biologic inputs just from the shorelines, just from there's lots and lots of dinosaurs known from the west part, in especially Alberta and Montana. So there's more productivity and yeah, more organic material kind of coming into the seaway from the west, and presumably from the east as well. So yeah, just being a shallow sea, having more nutrients coming in, yeah, it's probably one of the big reasons for why there's so many different animals and so much life here. Yeah, cool.
Okay, well, let's thank Aaron very, very much for a very interesting talk. Thank you.
Thanks very much. Yeah.